Hi, my name's Stuart Marcus, and I am the founder, CEO, and Chief Medical Officer of SonALAsense, a new company developing a non-invasive drug plus device combination to treat recurrent glioblastoma. Just to note that there are historically 3 drugs approved by the FDA to date: carmustine, insolubilized carmustine, and Avastin to treat recurrent glioblastoma. None of these are being used as standard of care. As a matter of fact, there is no standard of care medically for recurrent glioblastoma. And unfortunately, none of the advances in medical oncology which have occurred have benefited recurrent glioblastoma patients. Now imagine using the energy of sound to produce light within the brain that activates a photosensitive drug. The drug is so specific that it can reach glioblastoma cells anywhere in the tumor or in the surrounding brain and kill them. And if it could, imagine, this could convert glioblastoma, recurrent glioblastoma from a lethal disease to a manageable chronic illness. And that is a quote from Dr. Mitchell Berger, chair of our scientific advisory board, in his hopes for this new therapy. The therapy is sonodynamic therapy, which combines both ALA, aminolevulic acid tumor targeting, and MRI-guided focused ultrasound. Aminolevulic acid is FDA approved for the visualization of GBM tumors, as you see here. It's highly selective and has a positive predictive value of 98%, whether it's primary or recurrent glioblastoma. However, ALA is also FDA approved for killing cells through photodynamic therapy in dermatology specifically, and an investigational study at a clinical trial site in Germany, Professor Walter Schirmer has shown that ALA photodynamic therapy can kill recurrent glioblastoma cells in patients and enhance their survival. To do so, however, you need the apparatus you see on the right, which is highly invasive, extremely hard to develop and to standardize, and therefore it's still in that one clinical trial site, and commercialization is moot. However, it has great promise of photodynamic therapy. Now, we're using a device, MRI-guided focused ultrasound, to actually create the light and produce a photodynamic-like effect in glioblastomas. And the device is manufactured by Insightec, and we have a proprietary form of ALA which is going to be used together with the Insightec device under an exclusive licensing agreement. And this is ALA Sonodynamic Therapy, or SDT. ALA SDT has been shown to be have very positive results in animal models. And here, as a matter of fact, it's been shown to extend survival times after a single treatment beyond those of any other GBM therapy that's been attempted in these animals. So right here, for example, at the bottom Kaplan-Meier curve in the mouse glioma model study, you can see extended survival only with the combination of ALA and MRI-guided focused ultrasound. In the top Kaplan-Meier curve, which was obtained by a study that I did with as a SonALasense co-founder, together with the inventor of focused ultrasound for the brain, Claire Heinainen. And in that study, we were able to optimize the treatment with ALA sonodynamic therapy so that survival was greatly extended, and that's the light blue line, out beyond 60 days, which is unheard of in this particular tumor model after a single treatment. Now, I and my co-founder, Mark D'Souza, have had multiple successful exits from previous venture-backed companies. And our 3 most successful exits include Pellepharm, which had a structured buyout by Leo Pharma for $760 million, Lotus Tissue Repair, acquired by Shire Pharma for $325 million, and Dusa Pharmaceuticals, which was acquired by Sun Pharma for $230 million. Our scientific advisory board has key opinion leaders, Mitchell Berger and Scott Blotkin, well-known key opinion leaders, and Dr. Pejman Ganouni and Todd Mainprize, experts in focused ultrasound technology and clinical use. We are seeking $4.5 million in Series A for rapid clinical proof of concept The Ivy Foundation is non-dilutively funding our phase 2 clinical trial, which is going to be done at the Ivy Brain Tumor Center in Phoenix, Arizona. And we have a $10 million pre-money valuation to fund our operating costs. And this graph shows what this table shows what the use of funds is. So in summary, we have scientific reproducibility. The $4.5 million funds a clear path through clinical proof of concept, which you'll see by the second quarter of next year. And we're capital efficient because the Ivy Foundation is non-dilutively funding our proof of concept clinical trial. We have a strong IP position and exclusive Insight license agreement. And here's our contact. Thank you very much for listening.
Thank you, Dr. Marcus. Excellent presentation. Thank you. Does anyone have some questions for Dr. Marcus? You can unmute yourself or you can put it in the chat box.
Sure. How many of your prior investors in your other successful exits are participating in this round?
None, because they were all dermatology companies.
Playing with house money.
But actually, the co-founder and I have put in $250,000 of our own money to fund ourselves to this point.
Dr. Marcus, this is Dan Arbess. Listening to your presentation, I was thinking about Ed Boyden at MIT's work on optogenetics, which is, his focus is on neurodegenerative diseases, but has a lot of the same concepts. I was just curious as to whether you're familiar with his work and whether there's overlap with what SonALAsense is trying to do in GBM.
No, there's no overlap because what we're using is the selective glioblastoma requirement for ALA as a metabolite, right, rather than to do something as a marker or to signal something functional action within the brain. This is a metabolite required by glioblastoma for energy. It never stops needing it. Therefore, we're using its own requirement to be able to kill glioblastoma cells anywhere in the brain.
That's really interesting and quite comparable in a different way, parallel to what we heard from Arjuna a while ago on AG5.
Yeah. Arjuna is using an artificial molecule. This is a molecule that the glioblastoma needs, regardless of whether it's the first appearance. It doesn't develop resistance, and ALA accumulates regardless of the molecular genetic profile of the GBM. So that's very important for use in recurrent or resistant GBM.
It's great. Thank you.
You're welcome.
Any other questions for Dr. Marcus? Okay, well, everybody knows how to get to you. Thank you so much, Dr. Marcus. I appreciate it.